Look at a eukaryotic cell honestly, and there's barely any open space. It's partitioned into rooms. Watch what happens if I take every internal wall down. Every single molecule is still there, and it stops working. Nothing was destroyed, and the cell is gone. So the walls were never packaging. They were mechanism. 4 minutes on what a wall actually buys. Let's rebuild one room and go slowly. A membrane wraps a space. There's an inside and an outside. The point isn't that it holds things together. It's that inside can now be different from outside. That difference is the whole topic, and it pays the cell back 4 separate ways. A lysosome is a sealed sac of digestive enzymes, and they're built to work at about pH 5. The cytosol around it sits at 7.2. pH is logarithmic, so that's 160 times the acid. 2 nanometers of membrane hold that gap in place, and inside it, the enzymes shred waste beautifully. Dissolve the wall, and they land at pH 7. Wrong conditions, loosen the cell, chewing on your own proteins. Second payoff. A peroxisome deliberately manufactures hydrogen peroxide, which is a genuine toxin. The oxidase that makes it and the catalase that destroys it sit in the same sealed room. Spill it and it isn't diluted into harmlessness. It's now everywhere, next to every membrane and every strand of DNA. Dilution is not containment. The cell doesn't need that peroxide spread thin. It needs it never to arrive. Same idea, gentler version. One pathway builds a molecule, another takes it apart. Different rooms, both useful. Mix them and they undo each other. ATP burns, output is 0, and the cell can only keep one of the two. The nuclear envelope separates writing the RNA from using it, and that gap is where the cell edits it. Third payoff, and this one is pure arithmetic. Here are 14 molecules spread through the whole cytosol. They almost never meet. Now put the same 14 into a room 1/20th of that volume. Concentration is amount per volume, so each one is about 20 times more concentrated. Nothing was added. And reactions run on collisions, so 20 times each side is roughly 400 times the collision rate. Fourth payoff. Many reactions don't happen in a space. They happen on a membrane surface. So a mitochondrion folds its inner membrane into cristae. Watch the envelope. It never gets any bigger. The surface inside it more than triples. That's topic 2.3's problem, solved by pointing the geometry inward. Now the payoff the rest of the course leans on. A mitochondrion has 2 membranes, so it has 2 spaces. Pumps push protons out of the matrix into the intermembrane space. Get that direction right. That difference is the stored energy. Protons flow back in through ATP synthase, and the cell banks ATP. Dissolve that inner membrane, and the protons mix to evenness. No difference, no energy, no ATP. So here's the belief this whole topic exists to break. Compartments are organization, not mechanism. Take every wall down at once. Molecules present, 100%. That counter does not move. And all 4 payoffs fail together. Conditions gone, reactions colliding, nothing concentrated, no surface. The honest prediction wasn't a slightly worse cell. It's this. Same molecules, same enzymes, nothing works. Ask what the wall does, not what the organelle looks like. Compartments do chemistry. Which fixes the other mistake in this topic, treating an organelle shape as a fact to memorize. Here's one you've never seen. Inner membrane elaborately folded, sealed around a distinct inner space. Read the features. Folded means surface area. Sealed means it's holding a gradient or a local condition. So on the exam, never answer for organization. Ask what would break if you tore the membrane down. Want to step through this one yourself? Links below.